Hi guys. So today we're going to talk about genetics. And when we talk genetics, we need to start with this idea of heredity. And what is it to inherit your genes? When you inherit your genes, you're passing on DNA from the parents and their characteristics to the offspring. So whether you like it or not, you get half of your DNA from your mother and half from your father. And then those genes come together and make you. Well, how come we sometimes look more like one parent than the other? And so that's what we're going to investigate today. So what is a gene exactly? A gene is a unit of information on your DNA, like a little segment, that codes for a particular protein. And those are passed on, they're inherited from parents to offspring. So remember that in your somatic cells or body cells, they have a full 46 chromosomes. Those are your diploid cells, where you have one chromosome that you got from your mother and one from your father. And those are called homologous chromosomes because they're the same size, same shape, same traits. Okay? So you have two total of each kind. So on these chromosomes, you have genes for the same thing. Let's say, for instance, and this is simplified, but eye color. All right, so my mother has green eyes, and she passed down to me some genes that are encompassed in green eyes, and my father has blue eyes. So on the chromosome that he gave me, there is a gene for blue eyes. So both of them have the gene for eye color, but there's different versions of that gene, different colors. Those are called alleles, the different versions you have. So you can inherit a short gene from one parent and a tall gene from another. Those are both genes associated with height, but different versions of the gene that makes alleles. And then as we'll see with these alleles, sometimes one is dominant and one is recessive, and that makes some of them show up more than others. So for instance, and again this is simplified because really eye color is more complex in humans, but brown eye color is dominant to blue, and that's why you see more people out in society with brown eyes than blue. So when we look at our genes and our Punnett squares that we're going to go into, we have some terms and some ways that we lay things down. So the first term that we have here is genotype, and geno sounds like genes, right? This is the actual combination of alleles or genes that you have. And remember, you got one from your father and one from your mother. So they're represented with letters, capital letters and lowercase letters. And you'll notice they always get two letters for a particular gene. Those are representing your two different alleles that you got from your parents. So when we're talking about alleles, the combination you could get could be homozygous dominant. Remember, homo means the same. We've used this word several times in biology. So homozygous dominant means they're both the same and they're both dominant, which is represented by capital letters. So in this case, I did T's. It could be any letter, it doesn't matter. T's are nice because the capital and the lowercase look different. So if you're homozygous dominant, you would have two big T's, which you inherited a dominant gene from each parent. Heterozygous, hetero means different, like if you're a heterosexual, you like the different sex. So heterozygous is when your letters are different. Well, the only way for them to be different is to have one capital and one lowercase. That means from one parent you got a dominant gene, and from the other parent you got a recessive one. So in most cases, and it depends on the trait, you're going to show the dominant trait when you're heterozygous. But that means you could pass on the recessive to your kids. This is how traits sometimes skip generations, and why some of you might have traits that you see in your grandparents, but not in your parents. Right? So for example, with me, I was born with red hair. Both my parents have brown hair. Luckily, I have a grandmother who is very Irish, and she has red hair, because otherwise it would have been very strange when I was born that I had red hair. But it passed through, it skipped my parents because they had brown hair that covered up any red that was in there and showed up in me instead, because I would be recessive. So I am homozygous recessive for the brown hair gene, which means brown does not show up in my hair, and you can actually see the red that's there. So homozygous recessive, you got two little genes from each parent. So when we put what we're going to do is a genotypic ratio. It's always arranged in this order, from the most dominant to the middle guy to the least dominant. Or in other words, homozygous dominant, heterozygous, homozygous recessive. So that's our genotype, and we'll just literally count how many of each kind we have in our Punnett square to figure out that ratio. But you do have to memorize that it goes in that order. Now, phenotype is different, so think phenotype physical. This is the physical expression of your genotype, so basically what you're going to look like. It's arranged in the same order, though, depending on the kind of cross, we might not have a middle guy. So in complete dominance, that we're going to be talking about, this dominant gene in the heterozygous covers up the recessive like it's not even there. It completely dominates it. So this guy, if this was tall and short, and capital T is tall and lower t is short, big T would be tall. Big T, little t would also be tall. And you would have to have two little t's to be short. So you could be tall to short, which is dominant looking versus recessive looking. But there's some crosses that are not complete dominance, where the middle one is actually a blending of the other two. So in that case, big T would be tall, big T, little t would be a blending of tall and short, which would be medium, and little t would be short. So our phenotypic ratio will depend on the kind of cross, and we will go into that more later on when we talk about those kinds of crosses. So this is our Punnett square. When we look at our Punnett square, what we're really looking at is these are the parents on the outside. And so let's say this is dad at the top. This is dad's sperm. And so for this one trait, dad can make two kinds of sperm because he happens to be heterozygous. They're different. He can make sperm that are big B or sperm that are little b because that's what he has. So that's dad. Let's make mom. We're going to do Christmas colors. All right, over here, this would be mom. So mom and her eggs, she's also heterozygous. So she could make big B eggs or little b eggs. And so the Punnett square, what we're seeing here is their possible children. Each block is a possibility of having a child, and each one is 25% because it's like 4 quarters in a dollar. So what happens is this sperm comes down this way and gives that big B, and this egg comes this way and gives that big B. So if that sperm hits that egg, this baby would be big B. And in this case, big B is dominant, and that's going to be the color black if you're big B, okay? And the other possibility is that this egg comes over here and gets fertilized by this sperm, and now this baby is big B, little b, or heterozygous. But if it's complete dominance, this big B covers up that little b like it's not even there, and the baby appears black. And so this is where the genotype is different than the phenotype. Both these babies' phenotypes are black, but they have different genotypes. All right, now this big B sperm can come down here, and mom's little b egg, so they're heterozygous as well. So they're also going to be black. And then the two little b's come down. Now this is the weird little kid, right, that doesn't look like the parents. Both the parents are going to be black, and yet they can have a white baby. So this is how those traits can skip generations with those recessive ones. So this baby is going to be white, which is recessive. So for my genotypic ratio, it always goes in that order where I have, oh, this is going to get ugly. Okay, that's a big B. I went too far down. Okay, big B, to big B, little b, to little b. So when we're looking at our ratio here, I have 1 because I have 1, let me do this one. I have 1 big B kid, right? That's the 1. Big B, little b, I have 2, so there's right there and right there, 2 kids. And then little b, I have 1 right there. So it's a 1 to 2 to 1 ratio. So as a scientist, when I see these numbers, I know exactly what that means. That means I have 1 big B, 2 big B, little b's, and 1 little b. Now notice your phenotypic ratio is different. So phenotype is what you physically look like. So we always start with the dominant. So how many of my babies are going to look dominant, which is black? Well, I got 1, 2, and 3. 3 of them are going to be black. And then how many are going to be recessive? In this case, the one freaky little white kid right there, right? So that's how we do our ratios. And if you memorize that order, it gets pretty easy, but you do have to memorize it. All right, so let's erase all this gobbledygook before we move on to the next slide. Here we go. So what is a monohybrid cross? So mono, which we've heard before with monomers, means one. So a monohybrid cross is when you're looking at just one trait. Like in the previous one, we were looking at fur color on mice, okay? And in a complete dominance is when that heterozygous, that's the only one it really affects, so that big B, little b, all right, the heterozygous individual is going to look whatever the dominant trait is, in this case black, okay? And it's like this little b is not even there. It's been completely dominated. So most of the crosses we're going to talk about here today are these monohybrid crosses with complete dominance. And next class we'll talk about some of the crosses that don't work that way. So let's do a little practice here. We have some flowers. Red is dominant, so we're going to pick a letter. In this case, let's pick the letter R. So red is dominant, which means we're going to show it with a capital R. And white is recessive, and you use the same letter, just capital or lowercase. So white would be a little r. You don't want to use a W at this point. And then we have to look at who are we crossing. So we have to pick out what they are, and it says cross a homozygous, which means the same, and dominant. So I'm going to make my Punnett square down here. Hopefully this doesn't come out too hideous. Oop. There we go. All right, so we're crossing a homozygous dominant. So if dominant is big R and they're the same, they have to both be big R. So you have to know that homo means the same and that dominant is capital, and then it's not hard to pick it out. And we're crossing them with a heterozygous. Now hetero means different. Well, the only way to make them different is to have one be capital and the other lowercase. Now they're different, right? And so then we just fill in our boxes. So the big R's come down and across, so that would be big R, big R. This would also be big R, big R. This one would be big R, little r. And this one would be big R, little r. Okay. So these are my possible children. So some of the questions they might ask is, what percentage of their kids would be red? Well, if red is dominant and this is complete dominance, all 4 of my kids are going to be dominant, which means 100% red, right? Or they could ask, what are our ratios? So if we're looking at the genotypic ratio, I'm going to put a G here for genotypic ratio. And so remember, that's going to be the most dominant, which is big R, to the middle guy, or heterozygous, big R, little r, to the most recessive, which is homozygous recessive, little r. So if I look at my babies, how many of my babies are big R? 2 of them. How many are big R, little r? 2 of them. 2 dots. And how many are little r? That's going to be 0. And you do have to put that 0 in there because it is telling me something. So that's my genotypic ratio. All right, so now I'm going to do my phenotypic ratio. So phenotypic is what you're going to look like, and so the dominant trait is red. So we are going to do, for phenotype, how many of my babies are red? Well, in this case, all 4 of them. So we're going to put a 4 versus how many are recessive, which is white, and in this case, none of them, so it would be 4 to 0. There's only 2 numbers in the phenotypic ratio because we only have 2 options here, red or white, and that's the only ways you could look. So let's try this other one over here. Let me erase. Okay, so now we have seeds. So we have green as dominant, so let's make green a big G. Oop, there we go, which means yellow recessive is going to be a lowercase g. So same letter, just capital and lowercase. Then we're going to look at who are we crossing, but let me make my Punnett square first. All right, there's my Punnett square. And it says cross a homo, which means the same, homozygous recessive. recessive. So they're the same, but they're recessive, which means I'm going to have two little g's, right? Because G is recessive. Two little g's right there in one parent. And the other parent is homozygous again, so that means the same, but dominant. So dominant is big G's, and they're going to be the same, so I'm going to have two big G's. Two big G's. Then I fill it in. So big G comes across, little g down, big G comes across, little g down. I'm going to do I keep the big G's in front. I highly recommend that because it shows you that dominant trait. So if you get in that habit, it's a very good thing to do. So in this case, all my babies are the same, right? 100% of my babies are going to be green. So if I want to do my genotypic ratio, I'm going to do it over here where I have room. My genotypic ratio, it would be big G to big G, little g, to little g. So now I just look at my babies. How many babies do I have who are big G? None. So I'm going to put that 0 there and then 2 dots. How many of my babies are big G, little g? I have 4 of them. And how many are little g? That's a 0. All right, so that's my genotypic. Now let's do our phenotypic, what they're going to look like. So in this case, how many of my babies look green, which is dominant? So they have to have that big G. That's going to be all 4 of them are going to be green. All 4 of them are green, and how many are yellow? 0. Okay, because in order to be yellow, you would have to have 2 little g's. That's the only way. All right, so that's how you do one of these crosses that are complete dominance crosses. You really need to know what homozygous means and heterozygous and how to represent a dominant and recessive trait. And then you should be able to do any of these. So where did this all come from? This came from this guy named Mendel. Mendel was an interesting story. Mendel grew up poor in a time where people had to pay for schooling. And so his family could only afford to send him to like elementary school, but he was very, very bright. And so they wanted to keep educating him, and they were farmers, but their farm was doing okay, so they could afford to send him in the first place, which was good. But they couldn't really afford university for him. So what Mendel did is he joined the monastery. He became a monk. Because monks were all educated to read and write at a time where a lot of people were not, but they had to be part of the monastery, but the monastery would then educate them for free. So originally he wanted to be actually a science teacher, but he became a monk. So when he was a monk, he had a lot of free time on his hands, and he had some duties, like all the monks have certain things they're supposed to do at the abbey or the monastery, and his duty was to do the gardening. And while he was doing the gardening, he noticed some things about the pea plants that he was working with. He noticed that sometimes they had purple flowers, and sometimes they had white flowers, and sometimes the plants were tall, and sometimes they were short. And then he worked with them. And he started writing down all of his data, and he started breeding the plants very purposefully, which we'll talk about in a minute. And through this, he figured out this idea of complete dominance, and that traits were passed on genetically from parents to offspring. And so now Mendel is considered the father of genetics. He wrote all this information down and left it in the abbey, and then nobody found it for like 100 years. And then eventually they found it, and it explained all this genetic stuff. So really smart guy with a lot of time on his hands. So here's some of the characteristics that he looked at in his pea plants. The plant height, where the flowers were, if they were on the top or on the sides. The color of the pods, so there's green and yellow peas. They're all the same species. They're just different colors. What the pod looked like, if it was inflated or constricted, which means it was wrinkly. Texture of the seed itself, the seed color, and the flower color. So he's looking at all these things in his garden and writing down information. So here's a little diagram that shows you the difference between the plants. And all of these traits, luckily, were done by complete dominance. So they were pretty easy to predict once you notice the pattern. So what Mendel did is he used this thing called cross-pollination in order to study his plants. So cross-pollination is basically, he would cut the part that makes the pollen off the plant and then very purposely use a little paintbrush to transfer pollen from one plant to another. So he wasn't relying on insects or the wind. He was doing it very purposely so he knew which plants were breeding together. But in order to start his research, he had to get plants that were considered pure for their traits. So these are called the P generation. They are true breeding for a trait. That means they are pure, true breeding for the trait. They have the same version of the alleles. So they were either, if we were doing height, for example, they would either be big T, and they would be pure tall, or you could have some that were little t, that would be pure for short. And what he did not want is any that were big T, little t, because those are harder to start with, and you get babies that were a variety. So he did not want those. So what he ended up doing is breeding tall plant after tall plant after tall plant until all he got out of them was tall plants every single time. And then he bred short plants over and over and over again until all he got out of them was short plants. And that way he ensured that he did not have any of these heterozygous plants that he was working with. All right, so he's very meticulous about what he was doing with his plants. So here's a little diagram to show you like how he was transferring the pollen from one plant to the other, and then he would get the seeds and plant those and see what was happening. So these are different generations. So it turns out purple is dominant to white, which is usually always the case. White is almost always recessive. And so the purple covers up the white like it was not there in the heterozygous individual. So let's say we're using P's to represent that. If you were big P, you would be pure purple. If you were big P, you would be heterozygous, so not pure, but still purple. And if you were little p, you would be pure white, okay? So the white trait's recessive. It only shows up when there are two recessive genes. So we have the two pure parents, and you have a Punnett square on your paper that we're going to do this cross on. So I'm going to draw mine kind of small, but right up here. All right. So what Mendel did is he took his pure parents, and I don't like using P's because they look a lot the same, but I'm going to make, these are the homozygous dominant one, and then he bred it to a recessive one. So I'm going to make my recessive one super stupid tiny. There we go. So we can tell the difference because P's look the same. So he bred two pure generations, pure purple and pure white together. And so that's your P generation out here. Let me get another color. P for pure or parent. That's the P generation. Boop. That's short for generation right there. All right. And then what he got offspring-wise in here is he got children that were all heterozygous. Big P, little p. All right. Every single one of them was big P, little p. So color-wise, they all showed up purple, which told him that purple was dominant, right? First of all showed up purple. And this is called, inside here, these kids are the F1 generation. All right, F1 generation. Think of it as first family. That's the first family that was made. And the first family, they are all considered hybrids. So like a hybrid vehicle has a gas engine and an electric engine. These guys have a dominant gene and a recessive gene. And so they all are hybrids, but they all look dominant and complete dominance. Okay, and that's the first generation. So then he took those babies and bred them back to themselves, okay, which you don't want to do in animals. That's inbreeding. That's a big no-no. But in plants, it's actually kind of okay because they're weird. So ignore this part for now. I'm just going to do my next generation with the purple flowers for your notes down here. So then he took those kids that were all purple, but it turns out they were heterozygotes or hybrids. Oh, I should have stuck with the green. Okay, that's all right. So they're heterozygous, right? And bred them. And so what happened is they had this baby that was purple and homozygous dominant. This baby was also purple, but heterozygous. This baby was purple, but heterozygous. And then one freaky little recessive kid that came out white. And so that showed that there was some kind of hidden recessive gene in there. And so this generation is called the F2 generation, the second family, F2. And in the F2 generation, this popped back up. So when we're looking at the ratios of these things, I'll go back in your notes after we're done with lecture and showing you notes how we did this. When we look at the generations, this is going to be a very different genotypic and phenotypic ratio. All right, so this one, here's another example with tall and short. So these are the pure parents. So this one would be pure short, and this one would be pure tall. And when you bred them the first time, all 4 of these babies are going to be heterozygous, all of them. And then in the next generation, you're going to have then one, just like before, that is homozygous. 2 of these guys are going to be heterozygous, and then one freaky little short kid. And this is, like I said, how traits can skip generations, how you can have traits that your grandparents have, but your parents do not, because if they are recessive, they can literally hide in your parents' genes and then pop back out. Okay? So, let's go back to here. So here's the F2 generation again with the purple and the white. So pure generation 1. Then we got our hybrids down here, or heterozygous. And then in the F2 generation, you get 3 purple ones and 1 freaky little white one. So the white one came from recessive genes. So what exactly is this recessive? This is what Mendel figured out. The inherited characteristics are factors in pairs, like, for example, big T, little t, that's a pair of factors or alleles. And he found that one factor in the pair masked the other when it was present. So if this big T was there, it's like that little t doesn't even exist. They end up looking tall. And this only occurs in complete dominance, okay, which we'll talk about some other kinds later. So the dominant trait, if that allele is there, the trait's going to be observed. So you could be big T, and be dominant, or you could be big T, little t, and look dominant. Now, in order to be recessive, this one gets masked. It's not there. And so the only way you see a recessive trait is by having 2 lowercase ones. So there's 2 chances of getting these and only 1 here. And that means that recessive traits are rare, all right? They're not seen as often. Like I mentioned the blue-eye, brown-eyed thing. Blue eyes are recessive, brown eyes are dominant, and so you tend to see blue eyes less often in the population than you do brown eyes. All right, so we have these laws, and again, in biology, there's very few laws, but there are some laws with genetics because we can mathematically prove that it occurs this way every time. And so the law of segregation, to segregate means to separate, okay? So this is what's happening when we make our Punnett square. It's exactly what we're doing. We're taking the parent genes and separating them to show all the different possibilities. And that's what we have going on, these 2 different alleles, and we'll make this one the sperm because that's dad. All right, it gives you all the possibilities that you can get there. So it just means to separate, and that's exactly what we're doing already on our Punnett squares. All right, that's just another diagram showing you how we separate out the genotype to get all the possibilities. All right, then we have this law of independent assortment. We actually talked about this law a little bit when we did cell division, and we said that our chromosomes can line up independently of one another, and we said that an assortment of candy was a variety of candy, and that they get into where they're going independently of one another. And so we have another Punnett square that kind of shows this, that these are inherited independently of one another. So here we're going to talk about more than one trait. So this is not a monohybrid cross, but something called a dihybrid cross. So if we're talking height again, all right, so here's our genes for height, tall and short, and we'll do color with Y's, so we got yellow and green. You can have 2 traits, and you're going to have 4 letters. So this individual right here is going to be tall, because they have a big T, and they're going to be yellow, all right? So with this, if you have these 4 letters, you can get any combination of these letters. And it's a little bit like foiling in math, where you take this big T and you have to have one T and one Y. So big T could go with big Y and give us this in their sextile, or big T could go with little y and give you this in their sex cells. And then you could have little t down here. Little t could go with big Y and put this in your sex cells, or little t could go with this little y. And so more traits you have on a gene, the more combinations you could get. And they do this independently of one another, which gives us more variation. And we've talked about variation before and how variation is a huge benefit for organisms. So let's talk about our dihybrid cross. So where mono is 1, di means 2. So this is when we're looking at 2 different traits. And you can look at more than 2 traits, but once you get bigger than 2 traits, the Punnett squares get so big that your brain would just melt out of your head. So instead they use computers for those things to generate them instead of us doing them by hand. So this is the biggest cross that we're going to have to do. And it does show the law of independent assortment in that we can take this genotype, and let's say this is red is dominant and white is recessive and then tall and short, that we can get any combination. So the R with the big T, the R with the little t, little r big T, little r little t. And making those little lines kind of helps you to get all the combinations. So these are what could be in their sex cells. So we could take that then and put it into a Punnett square that instead of 4 squares, there's 16. And on top of here, again, this represents the gametes of the parents, their sex cells, and every combination. So if one parent is here, and we could have big R, big T, that's what we see right there, and then big R, little t, right there, and then we got little r, big T, and little r, little t. And so that's that parent. That's what they possibly have. And you have to have one of each, because when they come together, it should look like the parents, and if it doesn't, it doesn't work. This is where a lot of kids make a mistake, and they just want to put big R, big T, little t, except then you would have 2 R's in here, and that's not going to work out. This parent happens to be all the same, so everybody's going to get a big R and a little t, which is what we see down here. So how you fill this thing in, let me do another color besides green in case you're colorblind. Let me go black. All right. Is you put the 2 R's together and try to keep your dominant letters out front. So between the 2 R's, these happen to be both big R, so that's fine. So big R, and then I have the T's that I'm going to put together, and I'm going to put my dominant T first. Big T, little t. And so what this tells me, if big R is red, oh boy, I got to write words. This could look bad. Okay. Big R is red. And then little r is white. I'm just going to put a W for white. Okay. And big T is tall, like we've been doing. So that's tall. Okay. And then little t is short. And again, I'm just going to put an S for that. There we go. Okay. So that would tell me this individual is red and tall. And so I'm going to fill in the next one. It would be big R, little t. Now this one's going to look a little different, right? They're still red, but they're going to be short and red. And I'm going to do this one, so I'm going to keep my capital letters out front, make my life easy. Big R, little r, and then I'm going to do the T's, big T, little t. And this individual is going to be red and tall. And I'm going to do this one, big R, little t. So this individual is red and short. And these ones are actually going to be the same down here. I'm not going to go through and do them all with you right now, just because these are all the same. So it would be the same pattern over and over again. So that's how you fill in your dihybrid. And then they would ask you, like, how many of your kids are red and tall? And you would count them up. So here's one all filled in with our P's. So this is my parent right here. And these are all their letters, the combinations of the letters that they have in there, and the parent is the same. So you can see that inside here I can have kids that look a lot of different ways depending on which combination they have.